pathogen a disease-carrying organism? This could be a bacterium or a virus.
Well, considering our host organism took antibiotics and the pathogen hasn't been destroyed yet, this is probably a virus since viruses aren't affected by antibiotics.
But why does that happen?
It happens because viruses have different metabolic pathways to bacteria, which prevents antibiotics from doing their job.
The virus had to fight past the skin and mucous membranes of the host organism to enter the body. This is difficult because the skin is continuous and full of many tough, dry layers. It also contains natural organisms that compete with the pathogens and lysosomes, which break down the pathogens to try to enter the body.
The mucous membranes are also hard to pass through to enter the body because they're full of sticky mucus to trap invaders, natural organisms, or lysozymes that compete with the entering pathogen, and a pH unfavorable for pathogen entry into the body. I was able to fight past all these systems to enter the body though. I'm a phagocytic leukocyte, a pathogen-ingesting white blood cell. Phagocytes can easily move through the walls of blood capillaries to infection, where they can come in contact with pathogen cells with foreign glycoproteins on the pathogen cell walls.
Then the leukocyte ingests the pathogen through endocytosis, and a plasma membrane forms around the pathogen.
Finally, the pathogen binds with the lysosome, which contains digestive enzymes to break down the pathogen.
Antigens are either proteins, molecules, or parts of molecules found on me. Antibodies are used to remember pathogens. They are created by B cells to attach to certain antigens marking the pathogen for destruction. To make an antibody, a specific antigen is identified, and a specific B cell that can make that antibody will bind to that antigen will be identified.
Then the B lymphocytes will clone itself rapidly to increase the number of the lymphocyte, and the lymphocytes will begin antibody production.
Then the antibodies will circulate in the bloodstream to find their matching antigen. Then the antibodies will help eliminate the pathogen.
I don't break down all of the pathogen though. I keep a few of the antigens from the pathogen on my cell membrane to present to helper T cells. That way, they'll be activated and be able to tell the B cells what I need. And how does that stop me?
Once the helper T cells get in touch with B cells, the B cells starts to divide, and once that happens, you're going to be in a world of trouble.
What are you talking about?
When B cells divide, they divide into two types of cells, plasma cells and memory cells. The plasma cells secrete antibodies that attack the first infection.
Well, that doesn't matter because you'll eventually run out of antibodies. Actually, she wasn't finished yet. The memory cells might not secrete antibodies, but they live a lot longer and they'll be waiting for you in case you decide to make a comeback. So antibodies will be produced faster and in greater quantity.
Thanks to technology, researchers have developed a procedure that produces many specific antibodies of the same type. So, if the immune system doesn't react properly, this will help.
Yeah, it starts the injection of an antigen into an animal, like a mouse, and the spleen is taken after the animal goes through a primary immune response.
The spleen contains B cells that are fused with melanoma cells to become a hybridoma.
Yeah, and the hybridoma produces antibodies that have a pretty long lifespan, but vaccinations are just as helpful.
Oh really? How so? Well, we have the choice of getting a weaker version of you or a dead version of you.
What? That is not cool!
Yeah, this actually helps the leukocytes recognize the pathogen a lot faster. And memory cells will help the production of antibodies in case you decide to drop in for a second visit.
Yeah, that's right. But too many vaccines can overload an immune system, and you might be allergic to certain vaccines. But on the upside, it's a pretty cost-effective approach since it's cheaper than treatment. What's HIV?
It is a virus that attacks T lymphocytes.
Then they won't be able to communicate which cells need to be cloned to produce the correct antibodies. When they die, the body won't be able to fight off other pathogens and AIDS will occur.
HIV is transmitted through the exchange of bodily fluids. This includes sexual intercourse, unsterile syringes, and etc.
Some HIV patients are discriminated against in employment and other things.
Also, countries with inadequate healthcare have higher rates of infection. Oh no! Open wound alert! How are we supposed to fix this?
Platelets release proteins called clotting factors.
Then clotting factors activate the enzyme prothrombin to become thrombin.
Thrombin then turns fibrinogen into fibrin.
So finally, fibrin binds together with the platelets and the blood cells to form the clot.
We're so amazing because we have the capability to do things like clonal selection.
B lymphocytes rapidly divide to form more plasma cells, B memory cells, T memory cells, while T killer cells help defend the body.
So what types of immunities can we have?
Well, there's active immunity due to the production of antibodies by the organism itself.
Only after the body's defense mechanisms have been stimulated by antigens, though.
There's also passive immunity where we can get antibodies from another person,
which stimulates our active immunity.
Natural immunity is due to infection, which can sometimes be a small problem or sometimes can be fatal.
Yep, doctors give a vaccine to expose us to viruses so our bodies can make memory cells so we don't catch it the next time.